Hello, I'm Warren Minns. Welcome to my video on captive breeding programs and their effectiveness at stopping extinctions in wild animals. The Earth is currently experiencing a period of mass extinction, the elimination of huge numbers of animals and plants from our planet. To reduce the rate at which species are lost forever, captive breeding programs have been employed. Success has been achieved in a wide range of species, such as giant pandas and the Lord Howe Island stick insect. Captive breeding programs are very expensive, and they force unnatural conditions upon their captive animals, such as being in a confined space and restricting which animals they can mate with. They're not free to choose as in the wild, and they must mate with whichever animal is put in their enclosure. The problem comes when trying to release these captive bred animals back into the wild. Will captive bred animals, when released, breed with wild populations and help the species survive? In this video we will examine some research by Brendan Slade and his colleagues in the Department of Zoology at the University of Melbourne. This research will allow us to shed some light on this problem. The researchers took 11 male and 9 female mice from the wild grasslands of Australia. They took care to capture them from different areas to ensure they were not related. These mice were allowed to breed freely for a generation. Their offspring's DNA was tested, and these mice were selectively bred for 2 more generations. This ensured the 3rd generation mice were as unrelated as possible and represented the genetics of the founding mice equally. At this point, more mice were gathered from the wild. They were to be mated to the 3rd generation captive bred mice. They were split into groups of 3 males and 3 females of both captive bred and wild mice. They were all weighed, and they were placed in an enclosure where they were able to freely breed. DNA was taken and analysed from all adult mice and all babies produced. When the weights of the mice were analysed, it showed the 3rd generation captive mice, the black dots on the graph, were heavier on average than the wild-born mice, the white dots on the graph. This was true both at release and throughout the whole study period. DNA testing also showed that at release the captive bred mice had higher levels of inbreeding than their wild counterparts. The results can be seen in this graph. When released together, the captive bred and the wild mice produced 59 litters and 189 baby mice in total. 83% were either the result of a wild-wild mating or a captive bred mating. Only 17% of babies were as a result of a captive bred and wild mating. These results are rather alarming, showing mice preferred to mate with mice from the same background after just 3 generations in captivity. Living in captivity can change the way mice pick mates, preferring mice with a specific smell, behaviour or size. These preferences can then be passed on to the next generation, solidifying this behaviour in captivity. This means captive breeding programs could be of limited value to bolster wild population, breeding within themselves rather than integrating. To maximise the success of captive breeding programs, it's important to do these things. Maintain genetic variability in these populations. All breeding must be between unrelated individuals. The captive habitat, be that a zoo or safari park, must be as close to their natural habitat as possible. We must increase mate choice for captive populations rather than forcing mates upon them, and try and keep animals in captivity for as few generations as possible to ensure they integrate into wild populations.